So Anshika, we are
I'm
live.
audible?
Yes, sir. Yes, sir. You are
Yes,
audible.
sir.
Okay.
Anshika, we are live. Ritu ma'am, you can start.
Dr. Ritu, you can start.
बेटा रूम बंद कर दो इधर से.
Yes, ma'am. Okay. Ma'am, am I audible?
Yes, Ritu.
Okay. With great gratification, I, Dr. Ritu Payal,
आधा
from
गिलास पानी दे
Department
दो
of
बस
Chemistry.
मेरे को.
Ma'am, should I start?
Yes, Ritu.
Okay. With great gratification, I, Dr. Ritu Payal from Department of Chemistry, Rajdhani College, welcomes you all for the first session of 2 weeks online refresher course on natural sciences conducted by Department of Chemistry, Rajdhani College in collaboration with Teaching Learning Center, Ramanujan College under the aegis of Ministry of Education, Pandit Madan Mohan Malviya National Mission on Teachers and Teaching. I am delighted to inform you that Dr. Anil Kumar Mishra, Director of Institute of Nuclear Medicine and Allied Sciences, DRDO, New Delhi, will start the first session. Before pushing forward, I have a brief message for our participants. They are requested to post their questions in the chat box. Sir will take up the questions after the end of the session. Proceeding further, I take the honor to introduce our eminent speaker, Dr. A.K. Mishra. Talking about his academics, Sir has obtained Masters in Chemistry from Gorakhpur University, PhD in Chemistry from Banaras Hindu University. During his illustrious scientific career, he has held many positions like postdoctorate fellow at Université de Bourgogne, Dijon, France, research scientist at University of California, Davis, USA, senior scientist at Nantes, France, and visiting professor at Max Planck Institute, Germany. Sir has also worked as a consultant with the International Atomic Energy. Sir joined in Mars in 1997 and worked in the division of radiopharmaceuticals. He has wide variety of research interests, including molecular imaging agents, radiochemistry, specific MR construct agents, bioconjugate chemistry, and many more. In INMAS, Sir has contributed towards the creation of a state-of-the-art infrastructure for advancing molecular imaging and research center, which permits early detection of disease to pave the way for the development of newer strategy for personalized healthcare services for armed forces. The center has unparalleled facilities in terms of latest and highly sophisticated equipments and has received international and national acclamation and has been able to carve out a niche for itself with much successful international collaboration. Talking about his research career, Sir has published more than 300 papers in various international journals and has multiple US, European, and Indian patents to his credit. Dr. Mishra has affiliation to several professional societies. Sir has delivered a talk at the British Nuclear Medicine Society, United Kingdom, and has received several awards, including the prestigious DRDO Young Scientist Award by the Prime Minister in 1999. The list is long, but with these words, I request Sir to grace his presence on the online platform. Zoom is yours, sir. Over to you, sir. Sir, you are muted.
Unmute. Now I'm unmuted now. I'm audible.
Yes, sir.
Okay.
Yes, sir.
So very good afternoon to all of you. Ritu for nice introduction. Aniska for inviting me along with Mallika Oja to deliver this talk in refresher course. What I would like to take you all that how journey one can start being from any discipline. When we talk about any discipline, the most important thing comes in my mind that artist turns to be Nobel Prize in medicine and physiology. It was a very beautiful story about one of the greatest scientist in neuroscience that we talk about, the Cahill, that he was a very rebellion teenager and his father was not very happy with him. And he asked him that to go and do the shoemaking and the job of barber. So he started that doing repair of shoes and then working in salon to do the hair cutting and all these things. Then slowly and slowly he decided that I will become an artist. And that was his passion. And he was so good in visual aesthetics that whatever the design he made, either drawing he did at that time, it has become the hallmark for the future in scientific activity. So anything you talk about the frontal cortex, either you talk about neuronal networking, either you talk about any neurons, either punch that how they fire it, that was designed and developed by the none less than the Cazal, that at that time he drew the structures and design and all these fine arts that we talk about. The finally to fulfill the dream of his father, he pursued the medicine. And again, he was the topper in his college to get the one of the best, the student in the medicine. And it's not only that, he was just such a great philosopher, doing a lot of his sports, always, ready to help to anyone. And that was his character at that time. So anyone that who is pursuing any discipline of social sciences and natural sciences is very important to pursue with really great enthusiasm, and drive should be in such a way that you should be very solitary, not that you are very shy in nature. And then finally he became the one of the best doctor. He went to the Cuba, and after that, when he returned from the Cuba, he was suffering from malaria, and then he took his last breath. So you just look at these two guys that always I admire them, to seeing that the Golgi and Cazal, they really fetched the Nobel Prize at that time. And then you can imagine that his career started with making the shoes and being a barber. So I really, all credit goes to this type of students. And then finally they turn to be very nice teacher, either wonderful teacher. They take us to a domain where you can really learn from them, from their biography. So I suggest that whosoever is pursuing in any type of any discipline of the natural sciences, either in social sciences, either humanities, they should certainly read all this, that whatever they have done, and then how nervous system and everything function about this. So just look at these two diagram that I have put for both of you, to all of you online that is attending the classes, either the person who is coordinating it. This is the hallmark, that how neurons, they are connected to each other. The design that was made such a long time period, and that is still the textbook. So imagine that what he was doing and how neurons today fires, and then every nervous system, including from retina to hippocampus, is so nicely controlled. So this is the beauty about these two gentlemen, and from where life and discovery starts, and then people, they start working on this one. I put this slide to know that whatever we try to see from outside, either what we try to see inside, that what is happening. And that just look at this, the book that always we refer about, that species spanning approach to medicine, that what we select and to say that it mimics that what we are going to do it. I have put this slide, either this cartoon in front of all of you, is that we learn from this cartoon. We learn from these slides that we have displayed here. Why I have put this slide for all of you is that Barbara and Etterson at that time in 2012, they were discussing about that, what makes them, they are so happy and they are playing the card, and then you link with the Cazal, that neuronal system, and then everything that they connected, and we are in a good shape to perform anything. So reason being is that showing this slide, that we have to preserve the nature, we have to learn from the nature, and they are the best teacher for us, and they also, they sacrifice their life for us to do the research and to move forward. So keeping this one, I took a story from the one of the movie, and she was one of the best teacher, was known to do the teaching. And then finally, I'm sure that many of you have seen this movie like Alice Steele that we talk about. What was happening with this professor when she was teaching? Most of the time, she will not recognize, either she will feel something, she start teaching mathematics, she will disappear, she will start teaching something else. So this was a very inherent problem, and family slowly and slowly they discarded us. They were thinking that something wrong with this Alice, and she is not able to really communicate, either to attach with us. So I'm looking at this picture, and by looking that she's so healthy and everything is very nice, but whenever they were giving some tasks, something was wrong. So if we connect with this one, a vibrant, accomplished college professor disappears in front of her friends, family, herself, and turned out to be that she was really a severe patient of Alzheimer, and slowly destroys her mind. So putting this movie, either putting this novel that was written by at that time, and then they realized as a film by Richard Glazer, that they were talking about that some person who is very healthy, doing very good, but when it comes to perform a task, either to do a stimuli, she was always lost. And that was the reason that such a wonderful professor, and she was not having space in friends, in family, and herself as a Alzheimer person. So it makes us that being a scientist, either being a social scientist, either being a teacher from any domain, what could have gone wrong, and where we can really interfere, either really we participate to decode it and to find out a solution for such kind of malady, either such kind of disease that we talk about. And that doesn't go till only this place. When she visited the doctor, and doctor told that your entire family has to go for medical screening, it's not only you. Then she got more worried, and then she narrated entire story that you guys must watch this movie to her daughter. And she told that what the hell you have been talking, that I'm suffering from some disease. And finally they went to visit the doctor, all of them, and that turns out to be that it was a really genetic problem, that all members of the family, they will suffer from Alzheimer. So here it comes out that a kind of screening system, a kind of system should be very robust, that anything goes wrong to anyone working in any domain, we should be very, very curious and very cautious, and we should give our 100% to see that that person is taken care. So what was the story behind is this, that we have the brain, we have the distribution of all the design that we were talking about, the Nobel laureate artist turns to be Nobel Prize winner in physiology and medicine, is the electrical circuit of the brain. If something is broken somewhere, like the line he has drawn, and that line is still today, most of the people in zoology, botany, in chemistry, in neuroscience, they teach this diagram that we talk about. The question was that where it is broken and how it is broken and what is coming out on this one. Here, chemists, they try to see that can we find a solution? Biochemistry, the title that you were talking about, that why biological system is not performing the way it has to perform. Then question comes that then certainly some chemical locha is there, either some chemical disturbance has taken place. And to find out this chemical disturbance, there are many ways that we have to do this. And when it has to be done, how it has to be done, what should be the pathway, at what time we should interfere is very important. Reason being is that whenever we try to see that some critical point to find out that some problem has taken, and look at this diagram that I'm talking about, that any neurotransmitter fluctuation takes place in the case of Alice that we are talking about, means already some neuronal architect has started changing. Neurotransmitter is started behaving either upregulated or the downregulated. It doesn't happen when you are 50 years old. It happens when you are very young also. Because we introduce many type of insult, either we smoke, either we drink, either we do any kind of traumatic brain injury you talk about, either some type of insult has taken place, and that we never came to know, only when we came to know that the disease manifests, and then we are in a severe problem. So look at these two diseases, either you detect as early as possible, either you detect when it's very severe, and that is not the solution that being in science, in natural science, we should work. So here is a message to everyone working in any domain of natural sciences, either from biochemistry, chemistry, geography, geology, history, any, because we have to see the behavioral study. We have to see many, many parameters to say that, my friends, why he behaves like that. So the question comes that we have to really design a robust system to find out that if someone is not able to perform this task, it means that which molecule has changed, which polyamino acid has destroyed, and which type of metal ions are fluctuating. Either you can really classify it as a neurotransmitter, that two types that we have got, either excitatory, either inhibitory, that we talk about. But then question comes, being in a chemistry, that what to synthesize, what to build, to see that non-invasively that this person is suffering from a type of disease which is very deadly, and then we are being really ignored in the society. So we try to develop a stimuli. We have the monkey model. Then we try to give a task on the computer. They perform it. When they perform right way, they get the fruit juice. They don't perform in the right way, they get nothing to drink. So then they motivate themselves, because they are getting incentive. When they get incentive, then try to recognize the target in a nice way. And the similar way, when you are teaching, students are not performing very well, is suggestion that you should try to see that what type of incentive, what type of motivational, either motivation that you can provide, so he is a person who does the correct things in a correct manner, in a correct time scale, rather it is too late. So look at this one, is not something that big, something that it cannot be decoded. Either is we can say that is a very complex chemistry, biochemistry, and the social sciences. It's just starting from one of the amino acids that we talked about, tyrosine, and then you make all this neurotransmitter. Either you talk about norepinephrine, you talk about dopamine, and then you talk about epinephrine. In between, all the chemical process takes place, and then we have to see, we have to find out a mechanism at which stage of this entire process of synthesis of any neurotransmitter we can come in the picture to see that this basal ganglion, either this type of ventricles, they are completely open. Look at this one, this structure that here is, that ventricles are in a different size. Then it gets enlarged when we say that mild Alzheimer's disease, and then look at the last one where we say about the severe Alzheimer's disease. So this type of images you have to get only one way, when you do the postmortem. When you do the postmortem, either you have to do a very good sequence to see in MRI today that we have got the system to say that your ventricle is really widened, and neurotransmitters, they are leaking out of this, and they are not intact, and they are performing the way they have to perform. So you see that we try to take the tyrosine, we take the dopamine, we try to level it, and after leveling, we try to see that what type of changes takes place. So we try to do a lot of chemistry in this one. I will not go in the complete detail, but if anyone has question, certainly I will be very happy to answer it. So what we do is that we take some analogs of the tyrosine. It mimics that one in such a way that its biological half-life is very strong. Then the chemistry done in such a way that you can put the fluorine, either you can put the silicon in such a way, in the both type of nuclide we talk about, either unstable radionuclides, either stable normal nuclide that we get the natural nuclides, and then we are able to see in a nice way. So a lot of synthesis that we do it, and after synthesis you can see that we do the modelization. We try to see that you are able to see that it is binding to the receptor, that receptor has to be seen. And you can see that very nice 3D picture in tomography, that the compound you have synthesized, it goes to the ventricle and basal ganglia. And if there is any problem in this one, then certainly you can interfere. Either you can just see that what type of management can be done for interference, either to put the drugs that is required. So this is the one example. And then finally it gets translated to the human application, where the person is normal, either person is suffering from Parkinson's disease, either is suffering from the Alzheimer disease. So look at in my right hand side where I'm putting the cursor here. This is the basal ganglia that we talk about, like kidney that we see, either beans that we talk about. If there is any damage, either any change, as early as you can see that in this scenario where is unhealthy, that it means this is broken. This not enough neurotransmitter is being produced. Similar wise, you can see in the entire panel that how is the normal, Parkinson, and then you compare up the many types of the tracer that we design at our center to see it. So you see the story that it starts from that I'm happy, I'm suffering from something, and then intervention of a chemist, either the biologist, either the social scientist, come together, and then finally we come to a conclusion that this person can be diagnosed and treated, which is the very important for all of us. So when we talk about this portion, then we talk about the another portion that we are very rich in biodiversity. Ecosystem is very good, provides very nice services. If there is any perturbance in this ecosystem, then we lose. So how to really maintain this ecosystem? So our cultural, our behavioral, our social scientists, our natural scientists, science from the different branches, they come together, and then certainly we see that we maintain the ecosystem. What I would like to take here that the life and biodiversity is so important that we can extract many things out of it. And if we miss somewhere, then certainly we are going to play with the nature. And that it playing is the nature means that we have a different type of outbreak, either disease that we talk about. The best example that you guys have seen in this pandemic, that the outbreak of this COVID-19 virus we talk about. So you look at the bat, we look at the fish, you look at the animal, you look at the mosquito, they are friendly, but at the same time, also we have to see that what we have been destroying, what we have been communicating. So in this, I took one example of this one, that if you want to really see that our ancient life is maintained, to recapitulate it, either some lesson has to be learned that the species are going to extinct, where any bad communication is coming, is destroyed in the beginning before it reaches to you. So we have lost many flora, many species that otherwise this communication could not have taken. It could have been destroyed very nicely. Curing a cancer, it means that if you are not able to cure a cancer of a person, which is very important to this country. If I give one example of our defense minister, Parikar, if he died of cancer, it means that we were not really prepared, either we are not able to really protect our citizen from any type of attack that we talk about, the cancer and the pandemic. So we have to develop the marker. We have to develop a very strong social networking to do the therapy in a best way. And that is very important for any habitat to survive and to really explode, which is the cracks of anything that we talk about in future research. If you read this paper of Welter and we that published sometime back in 2015, you will get that how if we play with the nature and then finally it becomes the pandemic. That's very important for us. This slide I took very specifically for all of us. To synthesize in the lab this type of compound, it takes years and years in multi-step. If we have got very nice fauna and flora, it takes just one thing, that putting two things together and you get a beautiful compound which is applicable for many things. So taking from the nature, where you say that biosynthesis is taking place. Who does this biosynthesis? It is either done by animal kingdom, either plant kingdom, and that becomes the resource for us to use it for different application. And you can see that how nicely they have introduced the lipid, how nicely they have converted one of the serine to a dihydrosphenazine compound that we talk about, how lysis takes place and you convert to the one of the substituted amine in such a nice way, even such a systematically defined chemical pathway. In the laboratory it will be very difficult what nature does. So we have to be very smart that how we can protect this nature. I take one example in this one case study. We talk about this cholecystokine receptor, and this is available in plant kingdom, it is available in animal kingdom, and this is the one of the peptide, one of the protein that we talk about. They are very important for human. either you talk about the brain tumor, either you talk about the prostate cancer. If this is not in the nature, then certainly you cannot derive the medicine. Either you cannot learn that how to design these molecules for the clinical application. So what we have taken the lead from this one, that we do a lot of heat and trials in this one to isolate it, from where to isolate it, how to characterize it, to know that which amino acids are most important biologically, and that you have to plug and play it very importantly. Just look at this one, this plant extract that we talk about, that the case studies I have taken from aspergillus, and from where you take this aspergillus, you extract this aspergillus, and this aspergillus, that is such a beautiful compound. Looking at the structure, it shows like tryptophan, you have got glycine, you have got some of the phenylalanine, you have got some imidazole ring, and you break it. Once you break it, you can synthesize such nicely. And this compound is being used for many applications for human health. But what I want to say is that we always try to play with the nature. Look at this one, such a beautiful species that we have got, and then is very good for many things, that whosoever is they eat seafood, either they preserve it, either they take many kind of things. What we do is we always wanted to increase the cultivation of all these things by doing unnatural way, either by introducing some of the toxin which is not very important and that is very dangerous for us. So you look at that wild salmon, if you talk about, where we have all this toxin that is very less in concentration, like we talk about 4.8, but when you do the farming of this one, so we introduce a lot of toxic material. We wanted to have the very high yield of this one, a very weight that we can get it. So all this by doing the farming of this fishes, you can see that you have introduced almost 9 times of toxin in any scenario that we talk about, and which is very dangerous for us. But you just look at this one, that what we synthesize in the lab, the peptide that we are talking about, you were able to get from aspergillus, and you are able to get from the fishes, and then you see the concentration of these and the binding constant of all this that we are working on this one. So it's very important for us that how we preserve the nature, how we get this peptide from the nature, that is very important for clinical application. So not only this, that we have to destroy that many fauna and flora, but it's very easy to extract from this one. But at the same time, you cannot destroy it. So one of our colleague from a medical center of biotechnology, we work together and we try to see that this compound is also known in the literature by many companies. Then we came out that we can synthesize this molecule, we can evaluate this molecule, and very nicely we can demonstrate that this moiety can be very useful. These compounds are very useful for clinical application that we were getting from natural resources. So what we do is that nature gives us something. From nature we derive the molecule which is essential for us, and then we try to mimic it and to synthesize and to prove the efficacy that whatever that we had got in the nature is equally either better, either some compromised biological activity that we get it. So look at this one, very nicely synthesized. You look at the binding constant, so it's very nice, 0.63 nanomolar concentration. Half-life is almost 100-fold. It means if I'm injecting to any human being, either any scenario, it can reach to the target if a very longer biological half-life, it is not destroyed, it is not killed, it is not digested by enzyme, and then you can do many more chemistry to see it by using different type of non-invasive imaging. Look at this one. So we started from aspergillus, we started from the nature, we started from fish that we talk about, salmon, and we are able to build a molecule which is today utilized to do a kind of imaging which is very important for the medullary thyroid carcinoma. I took this slide reason being that in institute there are many thyroid cases comes. It is very useful for small cell lung cancer, and the most important that we talk about the gastrointestinal tumor. So it's a beautiful translation from taking the lead from nature and then to translate that how you can build this molecule can be used for the clinical application. So this is the one example I took it. Since yesterday I was discussing with Malika, she told me that the people from the geology group will be there also. Look at this one, such a beautiful. The first time this color was isolated from geologists. and they got this compound, and you can see the painting on my right hand side that they used to do for the painting is none less than this is called petroporphyrins. And this porphyrins, everyone knows that in our bioinorganic chemistry that we must be teaching to everyone. This porphyrins iron, and then it binds with oxygen for the transport at the function of different electropotential that we talk about. Such a nice protoheme that we say etiopropyrin nucleus, and nature does in such a way that if you look at that, you can do the lipidization that can cross the blood-brain barrier for different application. You can enrich with the nutrients, minerals, everything. And these molecules, if I have to synthesize in the lab, it will take years and years and years. So all the geologists, the group of the geology department, if they go and collect something like this that shows beautiful color, they should not really leave it. Bring something, do the extraction, and then you can see such a beautiful molecule, which is of very high important for photodynamic therapy as well as for the clinical application. And this was petroporphyrin was discovered by the French guy, the Corwin, that in 1960s, and that can be found in rock, cells, and sediments that we talk about. I'm showing this slide is that, again, I took an example from the nature, and by taking the example from nature, we have to build it, because we cannot certainly go and keep extracting, that will kill the time. So looking at the structure, looking at this one, then we try to see always that if I get something from the nature, I have to characterize it. Characterize in a way that by using different technology, we talk about that we use NMR, we use mass spectrum, we use UV spectroscopy. So many technology we use it. I have taken this one, that even if you get a molecule from the rock, either anywhere, even if it looks so bad in the spectra, there are many ways, many sequence, many contrast, many lengthenide reagents that you can use, and then you can really resolve the peak in such a nice way. So I want to show, if you look at in my left hand side, that one of the picture of A, where there is no splitting pattern, and look at the other side where you can see very nice splitting pattern of this spectra. And then finally it get translated in human application. You can even see each neurons, each networking that was drawn by the Castle at that time. And then you can do the anisotropy of this one, that is called fraction anisotropy, and you can make the all tracking of the fiber, in which direction it is going, it is broken, it is not broken. So then how it is possible to see that such a spectra which was not beautiful, we are able to see in such a nice way. Then the spectra that we are getting in human, it was not so beautiful. Now we can see such beautiful spectra, and that is much more informative that we talk about. So look at this. What we do is that everybody must be teaching in your classroom that you have got the gadolinium is such a beautiful elements in periodic table, highly paramagnetic, having excellent property to change the coordination number, and you can design very nicely. But problem with this one is that it's very toxic in nature. So to avoid the toxicity, what we do is that we design some chelators. The chelators bind it very strongly under physiological condition, so it doesn't metal is not leaked out. And that is the important for doing any kind of system that you want to develop that can get translated. They can be used for some application that you have thought about. But just taking the lead again from the nature, that you look at the crab. Crab is a system that holds very strongly the object, but at the same time, they have got the sensor, and that senses that what I have to sense in which scenario, and then accordingly, I can change my coordination number. Either I can change my property to become smart, and then to give the information which is required from medical point of view. So this is the designing of molecule, taking the lead from the crab, and then finally we are able to really see all this, spectroscopy to inversion recovery to see that at what time scale it is working. It is working in the millisecond time scale, either it is working at a second scale time, which is very important because any changes takes fraction of millisecond and very instantaneous. Even you cannot determine the kinetics of the changes that takes place, because today I'm looking at Ritu, after a few seconds I'm looking at fraction of second at Aniska. So you see the changes in my brain, that how it takes place. And for that you have to design the molecule which is really robust at a different pH, and then incremental changes you can observe. At the same time, you can change the contrast in the fraction of millisecond. So you can say that if any change takes place, I'm able to track it, I'm able to see it. And that's why you can see today that whatever we get from the nature and MRI is being done, you can see the pre-dose, we are not able to see many things. But when you give this contrast, you are able to see many a small lesion that we say always metastatic lesion, either micrometastatic lesion, and then you are able to plan the treatment very nicely, very strongly, keeping in view that this can be very useful, that we want to work with the system. Another example, I took it from the nature, where many zoology people, they go at different places. You do a lot of, dissection of the frog and many type of fishes, scoliodon and anything. What you do is just you throw it, either you, after doing the surgery, either you don't preserve, either you throw it. It's such a important message here for everyone, that you look at the skin of this frog that is called bovina, and this bovina is not very unique. It is found everywhere, but in India also. But it's a screen. You just look at on the skin that you have got many peptides, and these peptides are so important that you can treat the prostate and brain tumor. So we took the lead from this one, and getting some isolation from the skin of this frog, that is mombino that we talk about. And then you just design it, because being a chemist, once you get the structure, you do the reverse chemical engineering, and then after that you design the molecule, and after designing you do all the parameter which is required to say that it is like a natural ligand that body produces, either human produces, either someone, by just changing some of the activity that we always talk about, the biological activity, either biological half-life. It means that it is not going to change its conformation, is not going to change its configuration, and it is also not digested by the peptide, either enzyme that we talk about. And then we design many molecules that I'm not going to talk about more in this. And you can see that once you design at the real time scale, you can graft the tumor, you can see it. And then once it is really approved, either validated in the preclinical model, finally you can move by doing the imaging for different type of tumor where these receptors are overexpressed. You talk about pituitary, you talk about medullary, lung, duodenum, ovarian, anything you talk about midgut, and these peptides are overexpressed. And that's why you design the peptide. I can show you in this slide that such a beautifully nicely picture you can obtain with this one, that in your left hand side, whatever the red and yellow that you are seeing is the tumor. When you do the treatment, the amount of the spots and the yellow color almost disappearing. It means that first to diagnose and to treat it, and that you get the lead from the nature. What a beautiful system that you can design and is being practiced in the clinic today. Last part of my discussion is that most of the time we use the copper, and then our ancestors, they were using it quite often, but we had last summer that we switched over to the all plastic and polymer, because it was very easy to carry, but also after effects that was also very bad to carry. So that's the thing, that why they were using this one. It was a logic behind that, why copper was used at that time. You just look at this, that the beauty of the copper. You talk about any type of disease that today occurs, even that being exploited so nicely in COVID-19, that they make many masks, many compound that can be used and to protect with this copper. But being in a chemistry, either being in a science subject, being in natural science, if anybody says any story, we have to really verify, we have to see that, and after seeing we can come to a conclusion that whatever ancestor that they were saying is true, and then it works the way it has to work. So look at this. I took just a simple electronic configuration of copper, and that also plays very important role in the life, because here it comes about social science. If you look at the copper 29, we always talk about 4s2, 3d9, and I say that, okay, my neighbor is happy, I'm not happy, it doesn't go very well. So you try to see that I'm happy, my neighbor is also happy, and that copper teaches us in a very nice way. So you can see that I'm half filled, one is completely filled, and both are happy. And remember, this is the exception in the periodic table you're teaching every day to your student. But what happens in this one is that you take all this copper, you get from wherever, in which form, and how it works to help many things that we are working on this one. Just I take a call because DG is calling. Good afternoon, sir. सर वो 2 बजे आएंगे, अभी हम लोग आपको लेने आ जाएंगे सर थोड़ी देर में, and then after that we have a small discussion and deliberation, and then small visit. We have to felicitate our it will be 1 hour maximum, sir, from 2:15 to 3:15, sir. Sir. Sir, sir. Sir, sir. Sir, sir. Sir, I will come to tell you, sir. Sir, sir. Sir, sir. I'm sorry, I had to take a call from my headquarters. So just look at such a beautiful, even you look at the copper, it looks so beautiful. If you maintain, you eat food, you eat different type of fruits, you use the bracelet, you are using the mugs, jug, and any type of utensil that you have been using to enrich it. So here it comes the chemistry that in which form you have to consume it. Is in copper 1, either copper 2. They play very important, and is a balance in this one. So if you talk about copper 1 in monoamino oxidase, where it is very important in the brain to work about this one. The copper 2 is basically some of the protein, they bind it, they again change in the copper 1, and they become very active. So by keeping this balance, you look at the periodic table. When you look at the periodic table, always you have been teaching the copper 63, either copper 60, whatever the stable nuclei that we talk about. But look at the copper, we have got more than 100 isotopes, and each isotopes are so beautiful. They can be used for different application. Copper 67 for therapy, copper 64 for therapy and diagnostic, and likewise you have many coppers. It means that whenever that we are copper utensil that we were having, that is very important that it is there, but in what amount it is there, it was not known to many of us. But now everything has been really decoded. We know that what is the percentage of by isotopic distribution, that we have got many technology to verify it. And then when we verified it, it's also very important to know that how copper is taken inside the body. You look at this, the binding of the copper, and then where you look at the methionine is holding one of the protein, and that protein comes out of the one of the receptor that we call CLT1 receptor and CL2 receptor. And these are the copper transporter where they bind very nicely copper 1 and engulf inside, and then finally it goes there. But being a chemist, we don't believe totally that if we take something and we send it, it will be reaching in the same manner. Here we try to see that we design the molecule which binds very strongly, mimics the nature that we have got, but binding constant and stability is very high. And that's why we always try to see that we have to introduce some histamine in that, we have to introduce some lysine in this one, and also to make a kind of semi-cycle structure which binds very strongly. And then you can have many structure out of this that can be used for holding the copper very strongly under physiological condition, can be delivered for human application. And that's why very recently we published the paper on this one, that you load the copper, do the preclinical, and then after preclinical loading on this one, certainly you can visualize in human application. With this, you can see such a starting point from the copper, and they were saying that the copper was being used for many type of treatment, and you can see that you can visualize it, and you can treat with the copper, and that is the beauty of the copper. That's why our ancestors, they used to have many things in copper, either bracelet, either chain, either having the mug, either drinking, putting the water to get the different isotope, and these isotopes are very useful for different application. With this, we have to be all biodiversity, being the scientists from different domain, being natural scientists, social scientists, we have to work together to maintain the ecosystem, to get the information from ecosystem, and then finally we can develop that everybody lives healthy, happy, and enjoy their life. Thank you very much for your kind attention. And now it's open for your discussion.
Thank you so much for your delightful talk and for enlightening all of us with your sagacity. Sir, if you give me permission, I can pick up the questions.
Yes, please.
Okay. So, sir, the first question is put up by Dr. Mukesh Kumar Saini. He is asking what type of ligands can be used for gadolinium binding? Can we use gadolinium-based metal oxide nanoclusters for the MRI purpose?
So it's very important that whenever we take the gadolinium oxide is a rock. So you have to convert in such a way that it becomes labile and it binds with the polyhedral system, either crown system. So it's very important for us, either to anyone, to convert it first in such a form that coordination chemistry can be done. Once it is a rock, you have to change, you have to bring it to some labile group, and that labile group means that either you come in the form of acetate, chloride, either some of the alkoxide, whatever you think in your brain, it comes out from oxide to convert to alkoxide. Then you do the reaction with any ligand. Ligand mostly we being a hard base, hard acid, always you have to choose also hard base. So looking at this HACCP theory, certainly anything like nitrogen, oxygen will be very important to hold this gadolinium very strongly. In any condition you think about, but you have to really see that minimum 6 donors atom, either 6 coordinating site should be there to wrap it very strongly. Otherwise, again, it will leak out.
So the next question is, which form of copper should be used, whether it is in +1 state or +2 state?
So you see that you take any form, the enzyme does the job, but to maintain the copper one, when you are doing any studies, it will be converting to the copper one for sure. Even if you take copper 2, it will be converted. So it's always better to start with the copper 2, because to stabilize the copper one for in vivo application is very difficult. Let system to do it. So copper 2 will do the job for biological system.
Okay. So next question is, for making vessels to be used in our daily life, which metal is better, copper or silver?
Just see that you have to see the advantage of the metal and for the what purpose that you are going to do is. For example, if silver you are going to use for some bacterial either something, we are talking at a different level altogether. We are talking about that if very deadly disease that we are handling, where copper has got different isotopes that you can do the chemistry for, like, for example, if I talk about the copper 64, it has got 70% beta negative and 30% beta positive on the point of view of the radioactivity. So imagine with one nuclide, isolated one, you can do this job. And then also you have to see that you can do some coordination chemistry with this one. With silver, you are very limited. You can only do with the crown ethers, either something like that, you can deliver it. But to get the many isotope, with the half-life also you have to see very important that if it's a very longer half-life, then also again we cannot go for the clinical application. So we always try to see that the half-life, coordination chemistry, availability, and what type of isotope that we can enjoy. So even if you make the copper nanoparticle compared to silver nanoparticle, I'm sure that it will have much more age over silver nanoparticle.
Okay, sir. So the next question is that we generally, some people have left side brain active and some have right side brain active. So is something to do it with the medication or some kind of treatment, or to do with that Alzheimer disease?
Can you repeat your question? Somebody has the left hand side, somebody has the right hand side, right?
Yes.
Hemisphere that you're talking.
Yes, sir. Yes, sir. So is this kind of active brain part has to do something with this disease or medication?
No, it's not like that. What happens is that, you know, for Alzheimer's disease, there is a called beta amyloids, either the protein that we talk about. If you look at the phenylalanine, that when it does the pi stacking, then it becomes a crystal. It can be in the left hand side or the right hand side. So it stops the communication because it becomes a crystal. It is not soluble in the nature. So any transmission has to go, doesn't pass it. It just remains there because of the crystal, and that doesn't allow to pre to post synapse any communication. So that has to be dissolved. Then anything to do with the neurotransmitter upregulation and downregulation can be done. So that is very important, that we have to see that the crystal growth should be minimized, and for that the dietary control, meditation, whatever you are talking, not to insult, not to take the things which is going to make a stacking, either precipitation, either crystallization of the protein that we have got. That is very important. So even in the left hemisphere, either right hemisphere, either the meditation is very important to avoid any type of aggregation of the protein, either the peptide, either the molecule amino acid that we talk about. Otherwise, the communication will stop, and then that's why all process starts for Parkinson, Alzheimer, schizophrenic, anything that we talk about.
Okay, so the next question is, is there any specific age group that is affected by this disease?
It's not. It's any, for example, old age, certainly above 60, they will have to have, because the neuronal growth is stopped after 40. So certainly there are more of the neuronal death compared to the growth. But what happens that if I start smoking from today, young age of 4 or 5 years, certainly I will come in the category of this disease for sure.
Okay, so the next question is Alzheimer's disease genetic? And how long do people with Alzheimer's disease live after developing the disease?
You see that this is not a deadly disease. This is genetically also, because I gave you one example of this Still Alice, that they were diagnosed. You just read this book of Still Alice, that movie, and the novel that is written by the French guy. So is it genetically also at certain extent that we have the tendency of developing this, but that can be avoided. It's not necessarily that if you don't intervene. For example, I'm diagnosed in the age of 60, either 55. So I should see that coming from my family, I should do a history, and then also I go for the screening. So they should not suffer that the time I was diagnosed. So this is the one thing. In Alzheimer and Parkinson, what is happening is that the tendency of hallucination, tendency of forgetting, tendency of sleepiness, all this will be associated with this one. This is not the deadly disease, but is a disease that makes you a miserable life. That's it. Because you are not able to have the enough neurotransmitter, you are not able to communicate, you are not able to move your leg. So it's many things associated with the brain. So one brain is almost damaged. Everything is damaged, even if I'm alive.
Okay, sir. So the last question is that, is Alzheimer patients dangerous for themselves or others?
Alzheimer patient, what does?
Dangerous for themselves or others?
Yeah, you see that what happens when you don't know what, because you forget about many things. So what happens that anything comes out, either, for example, if they are working, they are not able to control, they fell down. So dangerous is means that they don't know, they can be any nature. You are trying to drive me to the suicidal nature, but it's not really that is the suicidal nature. What happens that they are dangerous for themselves because they don't know where they are going, what they have been doing. They don't know it is a fire, it is a water. For them, it's very difficult for this, and they will walk through. So they are walking. If somebody try to drive them, that is nothing, even if you hang yourself. So they will do it. So it's kind of things that anything comes in their brain, they are not able to react, either to control themselves. This is the only thing happen. Suicidal nature is completely different, where there are the, is the deprive of many neurotransmitter like dopamine, serotonin, is not, they are not producing. So they become very depressed, and then kahani, the story becomes completely different.
Thank you so much, sir, for patiently sorting the participants' queries, and for being a valuable resource person, and for sharing your expertise with us. Now, without much ado, if you give me permission, I officially conclude this session.
Yes, please.
Okay. So now I officially announce the conclusion of these sessions and request the participants to go for the recorded lectures. All the very best, participants.
Anything, Payal, that I have to convey?
Yes, Anshika ma'am.
Yes.
Sir is asking any other thing that he want to convey, he has to convey.
Sir, thank you so much, sir, and thank you for sparing your valuable time and sharing your expertise with us. Thank you so much, sir, for being a valuable resource person in this faculty development program. Thank you so much, sir.
Thank you, Ritu. Thank you, Anishka. And I wish you all the best. May I leave?
Yes, sir. Thank you
Thank
so
you.
much.
Thank you.
Dr. Ashish, can you stop the live?
We can ask Ravinder to stop the live session.
Just